Yeah. Next time.
회장님 시작할까요? 정윤 교수님.
네, 모더레이터께서 알아서 진행해 주시면
좋겠습니다. Okay, so I think now it's 4 o'clock, so we should start our symposium today. Good afternoon, everyone, and welcome to KSCBFM, the online symposium today. And I'm very thrilled to guide you through this insightful seminar about optical imaging and manipulation of brain capillary flow. And my name is Mina Seo, and I'm a professor of biomedical engineering department at Seongkyungan University. And before we begin, I like to introduce Dr. Masamoto briefly. And Dr. Masamoto did postdoctoral work at 2003 at University of Pittsburgh with Seungji Kim. And then he went back to Japan in 2006. And now he became a professor at University of Electrocommunication of Japan in 2008, and he's now a full professor of University of Electrocommunication, and he is belong to Center for Neuroscience and Biomedical Engineering. And today's title is about optical imaging and manipulation of brain capillary flow. Without adding further ado, I introduce Dr. Masamoto.
Yeah, thank you very much. Can I share the slide here? Can you see it? Is that okay? All right. Yeah. And first of all, I am very honored to present my recent study in this community, and I want to say the congratulations for the next brain meeting in 2025. I'm really want to attend the brain conference. 2 years later. Okay, so most of my talk will be the very narrow range of the technical part to how to measure in the brain capillary diameter and the capillary flow based on using the optical two-photon microscopy and also kind of the image analysis techniques. The motivation here is for neuroimaging mapping. We know the viscous stimulation to the mice, it is always localized in the metabolic signal here, like almost the same size of the barrel cortex or neuron unit. But if you look at the hemodynamic signal here, you measure laser speckle imaging, we know the flow change is spread over the activation area. So why this happened? That is because we know the vasculature itself has a signal transfer from the active area to the along the vascular networks. So this paper nicely shows the vasodilation induced by neural activity first to start from this capillary area. This blue color shows the initially respond to the viscous stimulation, and later this dilation is spread to the higher penetrating arteriole here. This is a cross section of the cortex, and also spread to the second and third capillary here. So the signal spread along the vasculature, but we want to know how the signal first evoke the capillaries. So we know the local neural activity cause the remote vasodilation in chiral arteriole and also penetrating arteriole. This is a powerful way to recruit the blood from the pool of the arterial blood from on the top to the parenchymal area. So, but we want to know how this blood is spread along the capillary networks. It is important for mapping and also important for the physiological basis. And today I want to focus on first how much change in brain capillary diameter in response to neural activity, and then I'm going to talk about this change in brain capillary diameter, how this affect the blood flow itself. And finally, we will discuss about what else affects capillary blood flow. So the beginning, little bit, we want to share the basics of the brain networks in terms of the neurovascular coupling. So if we want to understand the mechanism of the blood flow regulation, we need to know the blood pressure distribution along these across the vascular networks. It is important to know how the flow is driven by the pressure rise. So this information is actually missing. We cannot measure directly the blood pressure distribution. That's a limitation of our current status. And then, but based on the structure and the flow information, the one paper shows the estimated how the blood pressure distributed to these along this capillary the vascular networks. So this is initially start from the pyle artery level that accounts for the 50 mmHg in the blood pressure, and that goes down to the venous side. This is a kind of a normalized length of each vessel. So basically this the changing how much changing on the blood pressure and along this vascular component. This is very important because this is a driving factor to produce the blood flow change. So this is here the 40 mmHg is almost the input part of the penetrating brain tissue. So this is showing the estimation shows the 1/3 of the pressure is distributed along the penetrating arteriole. And this is the highest blood pressure, this difference along compared to the length of the vasculature. So there's calculated that as the flow change is driven by the pressure difference. So if this is high, that means the effect is high. And we know there's penetrating arteries regulate mostly the blood flow in the tissue because of this highest pressure difference, and also change in diameter cause the force power of the change in the blood flow. But if you look at the data, this capillary is very short, small length, but the pressure difference is high. This is kind of surprising. So still capillary had a power ability to cause the change in the blood flow. 50% of the pressure is still localized in the capillary area. So we need to know how much these flow changes caused by the diameter change, or also in this area. I will focus on later this viscosity part. This is also the important component to cause the blood flow change. Even though this is linear half of it. This is a force power, but it's only fast power, but this is still an important component, and we cannot measure also the viscosity change. Okay, and to do that, we need to know the vascular structure completely. Usually we took the area volume 1 mm by 1 mm by 1 mm squares using 2-photon microscopy in this anesthetized mice. And we reconstructed based on the software. We customized MATLAB software to 3D reconstruct it. To involve also the 2-photon is somehow tricky because the optical signal is widely spread to the axis direction. So we need to normalize that component to accurately reconstruct the capillary networks. And then within this area volume, there's about 10,000 capillary is here. So we need to look at all the capillary diameter changing during the activation of the neurons. So this is the first step. We 3D reconstructed, and the center point was extracted, and then also categorized based on the diameter of each point of the capillaries. And then we calculate the volume in each vascular component. Red color shows the arterial, and the yellow color shows the capillary. Here is the diameter less than 8 micrometer. We define it, and also veins. This is over the depths. So we if we compare those volume of the capillaries, mostly the 50%, more than 50%, depending on the depth of the cortex, but more than 50% is composed by the capillary, and other 1/4 is arterial and veins. And this is our typical volume in the mouse cortex. To evoke the neurons, so we use the whisker stimulation also, and then we repeated the different wave frequency of the stimulation, including the resting time, and more than 80 times we repeated. And to measure neurons, we use the GCAMP expressing mice neuron expressing GCAMP calcium sensors in genetically. So here the represented the one slice image. The red is the capillary, and the green is the neurons evoked by the. This is specifically in the 4 Hz stimulation here, and then we measure within the volume how many neurons to activated in each stimulation trial, resting on 1 Hz, 4 Hz, and 8 Hz, and we counted the neurons, number of the neurons, and we compare these. Each trial by trial to organize, arrange the trials based on the small number of neurons to the high number of neurons activated. And then if we compare the volume change in the capillaries, so we can understand the neurovascular coupling basically. But surprising, if you look at the capillaries, this is a very small change, and also we cannot detect any significant the neurovascular coupling based on this area. So this is because the capillary diameter could be changed in. Please feel free to post any question in chat room. Yes, please. And then also interrupted here. So, and then if you look at this, because of this capillary may be dilate or capillary constrict, all together we included. That's also mean capillary is almost very small change. So we need to clarify and differentiate which capillary dilate and which capillary constrict. So if in capillary basis. So to do that, we need a software so that we calculate in each point to the capillary diameter. This is about, we have more than 6,000 points is in the here, depths of 100 to 300 micrometer of the, in this 250 micron of volume of in the cortex. And then mostly this blue color indicates a 5 micron in diameter. This is the consensus, right? Then if you look at the capillary diameters, so all together the user making a histogram, actually the capillary diameter at least and 8 Hz stimulation, slightly the number of this mean number increase, but almost same, right? And then try to clarify the separate where is the dilate, where is the constrict. To do that, we use the statistical method. We have 20 trials in each resting condition and stimulation condition. Basically, use these capillary diameters to compare the 20 trials variations, and we calculated the t values between the resting and activation in each measurement point. Then we found that we characterize if the t value is higher than 2, there's a red color indicates that point is dilated, and the blue color indicates a constricted. So basically, this green color indicates no statistical change. So that more than 84%, about 84% of the capillary location actually showed no significant change in diameter. And this is 6 animals is consistent, and about 10% is dilate and 6% constrict. We need to want to see where is this dilation to constriction. So if you look at carefully, they compare the point of dilated and point of that constricted, and compare that these distribution. This is a cumulative histogram. So this is a little complicated. But if you look at the resting diameter, those dilation capillary actually showed the smaller capillary diameter compared to these unchanged normal area. And the constriction point is actually higher value of the at rest. The baseline capillary diameter high, and that's constrict. The baseline diameter small, that's dilate. These are the data shows. So that maybe could change over the artery to the venous heart, but it is not true. So we compared the networks basis from the penetrating artery to the venules, then divided to the each capillary's location based on the distance from the arterial to the vein, based on branching point. And this is the mean length, and this is the mean diameter at rest. So that, as you expected, the arterial side is slightly higher baseline diameter, and this is a fluctuation level at rest. So they're basically similar, but the smaller capillary here in V5, V4 is the farthest from the artery to the vein. There's slightly higher variation at rest. And if you look at the dilation and the constriction, but basically the arterial side, venous side doesn't matter. So no much change in both those capillary, or is uniform change in the across the artery to the veins. So the change is small because of the could be due to the neurons, evoked neural activity is small during the functional stimulation. So we enhance the stimulation to using the cyanuric acid to cause a seizure. Actually here, G-CAMP activity is high. And then you can see here, as you expected, the capillary diameter mostly dilate. This is blue color is 5 micrometer, but this is red color goes up to the diameters. And then this is for animals. So during seizure, most of 46% of the capillary actually dilated, but not the remaining 54% still unchanged. But those dilation and no change, those capillary had the different resting diameters. This is also consistent with the result of the viscous stimulation. So the seizure caused the dilation. That is a smaller capillary at rest, 4.3 micron, goes up to the 5.1 micron during the seizure, but no change. Those are almost the same setting of the capillary diameter at rest under the activation setting. So then we all solve it by based on the capillary diameters. This is a blue color shows the resting capillary diameters, and green is viscous stimulation, and red color is during the seizure activity. So you see here the smaller capillary is more dilate, and the larger capillary has almost the same during the viscous and also in the seizure part. So if we calculated how much change in each category of the capillary at rest diameter, so you see here small capillaries dilate due to the viscous stimulation, more dilate in the smaller one, and it's also seizure. This is consistent. If you look at this carefully, this graph, so that means the almost the 7 micron diameter capillary at rest is no change during the seizure. So this is kind of, we call this is the upper limit of the functional stimulation induced in the maximum dilation. We thought this is the capillary's diameter maximum upper limit. So summarize here, how much change in brain capillary diameter in response to neural activity depending on these baseline diameter at rest, that the viscous stimulation caused the 84% has no change in our experimental condition, and 10%, 6% at dilate and constricted. So if you increase the stimulation, increase the neural activity, use the seizure, so still the 44% has no detectable change, but how about the 56% of the capillary dilate? And the message is here is this extent of dilation is basically dependent on the diameter at rest, and that causes most of the capillary has the this means that the smaller capillary is highly dilated, but the slightly higher diameter capillaries is shrink. So the upper limit is would be the around 7 micrometer, regardless of the arterial to the venous axis. This is our first part of my first part of the result here. So then, what caused this difference in the resting capillary diameter? This is a very important question. So how do change the brain capillary diameter, and also this change how affect the blood flow? To do that, I need to summarize a little bit the recent controversials. So you know very well about the capillary pericyte. Could be there's a regulator, master regulator, based on this Nature paper. But later, the other group, the American group, showed these are not capillary pericytes, but this is arterial smooth muscle. But it doesn't matter the name. It is also the vasculature still increase in diameter, but slow. This is the dynamics is very slow. It's different in the speed of the change. So this is a kind now consistent. Now and now the consistent to the using the optogenetics. So if we express in the channel lobes in the pericyte, so that we stimulate here the optics, then stimulate the pericyte to cause the vasoconstriction here. Sorry, yes, here the capillary diameter shrink with the channel lobes in the pericyte, but controls has no effect. And this change in capillary diameter, it's actually, you see here, very slow. This is a 1 minute time. And then also certain point, the constriction of the capillary cause the slowing the red blood cell flow. This is this is consistent with in our results. I want to show this recent paper data here. So we also use the channel lobes expressing transgenic mice in ND2 cells. ND2 is including in the pericyte, and here green YFP green color shows the individual pericyte here in the capillary and penetrating arterioles. This is vascular mural cells. And if we simulate these capillaries during due to the light, the blue light, here is light off and here light on the same location, and we inject the microbeads in this intravenously. And here, under normal condition, you see that all capillaries are perfused in the microbeads, 4 micrometer diameter microbeads. But if you stimulate these channel expressing pericyte, there you see here the microbeads is stuck in certain locations. So that means that activated channel eruption expressing pericytes could be shrink in this location. So try to understand more finely. We see the use the 2-photon microscopy here. The pericyte expressing channel eruption, and we stimulated blue light, and you see here, if stimulate these black dot indicates the individual cells, and activate the pericyte to stop these red blood cell flow. Here the capillary diameter of this section, they show that this brain stimulation caused the constriction of the capillary, a little bit slow dynamics, and then flow stop this diameter at this 5 micrometer diameter. And then after that, the capillary slightly dilate, then flow restarted. This is consistently observed in any regardless of the pericyte types. You know that ancestral pericyte, mesh pericyte, syncytial pericyte, depending on the location, the capillaries, this shape, all these pericyte is consistently activate. Only the difference is the baseline diameter. This ancestral is more arterial side, so diameter is higher, but mesh and syncytial is a smaller capillary in the venous side, and also they are around the capillary. So if you stimulate using the optogenetics, there's ancestral is a highly constricted 10%, and others are almost on the 5% in each. And then, but how about blood flow? The capillary flow is actually in arterial side is not stopped. This is a color indicates the RBC stopped or not stopped. The white color indicates the nose flow change. Then the x-axis is the diameter. So those the stimulate cause the 10% change in the capillary diameter average, but no flow speed change, no stop change in the red blood cell. But in the venous side, most of all venous side, these red blood cells stopped. So even though the same change in the capillary diameter, effect of the flow is different depending on the arterial or venous side. This is a difference in the last time I showed. How about chronic disease condition such? This is a good question. So we didn't try yet so far, but I have a similar data set later. I'll show you the and the other key question. Sorry, but I missed that. Capillary diameter change with any regional difference. We didn't try another reason. Only the somatosensory. Sorry, it's also a good question. But we need to know more. Maybe change depend on the depth. This is also we want to see in only layer 2, 3 we so far looked at. So and that is we illuminate over the all area, 4 areas of the brain. So we more local. If we stimulate more locally, because the upstream constriction of the cap in the arterial side also decrease in the blood flow. That is also contaminating in this case. So we use the 2-photon microscopy to stimulate in the cellular basis. So if you stimulate in this pericyte, and then we measure at the same time in the capillary diameter in these capillaries, and this you see the inconsistently this cause the shrink of the capillary at the location of the stimulated plate port. This is time course, and then this is only you can see the stimulated side, but the near location or far of the along the same the capillary, so does show no change in it. So the stimulation caused only the local area of the capillaries and at the localized and in this stimulated area. We also use the control mice, but that's not change in capillary diameter. So this is because of the due to the express activation of the pericyte. And this is also consistent with around the encasing mesh syncytial pericyte. But the venous smooth muscle cell has no change in that. So next we this is on the we know that this is consistent. The pericyte can constrict can manipulate the capillary diameter. But how this change in diameter affect the blood flow is important. So next we use the transgenic rat that to express the fluorescent protein in red blood cell membrane. So basically this green color indicates the individual all red blood cells. And the red color is we use a sulforhodamine. That's a marker of the plasma. And if you're lucky, yeah, sometimes you can see the black dot. That black dot is unlabeled cells. That is white blood cells. So we can basically use this one to how many red blood cell and white blood cell and also blood plasma. Is individually we can look at the dynamic color change. And this is almost real time. And then we use the high frame rate because the sorry for the noisy image. And if you look at the bifurcation here, capillaries, this is a conference part. The two capillary is they get together here and mixed here, and then red is plasma and green is RBC, and we calculate this 60 seconds, 1 minute the change in fluctuation of this velocity and the diameter individual capillaries. You see their red blood cell velocity is fluctuated over 1 minute here, but the small change in capillary diameter. That indicates the velocity change occur with independently of this diameter change. So you see here the very slight change or almost no change in the capillary diameter, no detectable change in your eye maybe. But the flow change is very dramatically and fluctuated. And if average all these main capillary and these daughter capillaries, diameter, velocity and diameter, the velocity is these goes to two capillary gets higher in this daughter capillary, but those mother capillaries that slow the velocity. But those three capillaries actually you see the almost the same diameter, is so again indicating that the speed and diameter is not always correlated. It's an independent parameters. And here another example that is bifurcated, divergent. This is a mother capillary and the daughter capillary, two daughters here, but the diameter is very different. But if you look at the speed of these individual red blood cells, so the main capillary is 0.4 millimeter, and these two is about 0.1 and 0.2, 0.15 and 0.2. So they're most similar. So but the diameter is very different. So again, this is not the flow speed in the capillary is not always related to the diameters. So another finding here is very interesting. So there is a red blood cells evenly distributed, divided to this point, and also this point, and this is under normal condition. So we can imagine that the one capillary had the red blood cell that caused the increase in the resistance. That means the next red blood cell get into the other capillaries. So this is under the normal condition. If the increase in the number of the red blood cells, what happens? So we use the kind of disease mode is if we restrict the waters, dehydration condition, that makes an increase in the hematocrit. So that means the capillary red blood cell actually is stuck in many capillary red blood cells here, and also this is kind of crushed into the, and sometimes red blood cell is fluctuated, which is going, so which is. So this is very interesting because the both capillary resistance is almost even. So then the small change in the single red blood cell doesn't cause the in the resistance. So that means the next red blood cell is get fluctuated. So another example here is a kind of car jam, traffic jam. You see here the many red blood cells get into this flow. So there's the ones maybe the beginner of the driver. You cannot get into the highway. So that's fluctuated, but it means that this fluctuation of the resistance is small, and that caused the little chance to get in this blood cell. So good. You see here now that this cell has get in. The next one coming here. So the very balanced. If this also the another input here, the single capillary, but this is a higher balance. We maybe stable, so that doesn't come into the red blood cell here. So these are all flowing the other way. Another examples. This is all the dehydration condition. And this is first going to the left side, but the car suddenly comes back. And what's happened? So here, black one, you see the big one. That's a white blood cells. Okay, so the red and is into the white blood cells that increase an increase in the resistance. Kind of this big track here, and then stacked here. So all the small red blood cells is stacked here. So it's always the capillary flow is balanced in the resistance of the single vessel. That is the reason why the diameter change not affected to the velocity of the individual capillaries. So summary here, it is important how in the individual cells is packed to the across the networks, because this is increase the resistance, cause the balance to capillary flow. Once the balance is broken, for example, dehydration, so make some route to the shunting cause that cause the other area has the little blood cells. So balanced flow due to cellular packing is important to maintain the resistance and the fluctuation in the capillary networks. And if unbalanced, only the single pathway could be the shunt pathway happen. So that this causes the little flow here. Okay. So take-home message 2 is the how to change capillary diameter affect the capillary blood flow. So its impact of the diameter is all depending on the across the networks of the artery to the venous side difference because of the due to the different pressure difference in depending on the capillaries in the network. And the constriction affect on blood cell flow in the venous side, and the spontaneous fracture. This is independent of diameter fractures. That flow change in the cell flow. This is the the apparent viscosity depending on the tube diameter, and this is theoretical part. And this if the small than the red blood cell size, the shrink, the increase in the suddenly increase resistance. This is because the cellular is stacked. So this is also we know this is independent of the diameters. So it's a kind of dissociated. And then we need to know this how this viscosity change, but this is not measurable. Okay. The capillary is almost not a plasma flow. It's always we need to think about the red or white blood cell flow. This is my message here. Maybe after 5 minutes. What else affect capillary blood flow? And I don't have the exact answer here, but I want to summarize a little bit the literature here. So the capillary, the flow is cellular flow is interacted with the endocellular cell surface layers. So if you listen, this report shows the microcirculation failure after subarachnoid hemorrhage is reversed by hyaluronidase. Hyaluronidase is dissociated to degrade the glycocalyx, the smoothing in the capillary layer, capillary surface layer. So diameter slightly increase after the hyaluronidase injection of the hyaluronidase, and then this is shrinked. This is smaller in the non-perfused capillaries, recover the capillary flow and increase the capillary velocity and also pulsation. So basically this paper indicates the endocellular surface layers important component of the capillary flow in the microcirculation. And other paper also shows the after in ischemia. So these black color indicates the stored blood cells, and this TPA causes the dissociation, dissolve the blood clot. So then they recover the mostly patent, and then but there is leukocyte is stacked. So then if we use the anti-leukocyte drug, so this is improved the flow change more at only the later part here. You see the increase in the CBF after the injection of the anti-leukocyte. Also, this is indicating that the important in white blood cells in the cerebral microcirculation. And this is also happened during the spontaneously. This is all example of the stroke, but this is aging people. This is spontaneously sometimes shrink and stacked, and this causes endocellular degradation in later part, and this cause the number of capillary decrease. So they estimated age dependent density of the capillary change due to the spontaneous leukocyte, due to the spontaneous obstruction of the capillary flow. This is very important. We have to enhance the capillary flow. Maybe time is 2 minutes. Okay, I wanna slightly skip this. Okay, this is already more steady state condition. We use a chronic hypoxic experiment, how the brain capillary is adapted during the 1 month of the hypoxia. You see here the capillary dilate. This is different of the functional stimulation. At that time, I showed that the 7 micrometer is the upper limit, but this is morphological change. So cause the 8, 10 micrometer. Almost 1.5 times increase in the capillary diameter during this hypoxic exposure. So But this is not so effective to shrink the diffusion distance. Only the 1 micro changes. So you can not see the very difference. And this is the color indicates the distance from tissue to base cells. So even though the capillary dilated, but almost no little effect on the tissue to capillary distance. So they need to increase the networks to improve the oxygen diffusion. So this is an example of the new capillary appear here, you see here. And then this is hypoxic day 8 for 11. So you see the new network here. It's known here. And then at that time, we found that 10% of this sprout is actually disappeared. So what's happening? So we look at now we this is a kind of preliminary result, sorry, but now we shifted to look at the microglia here. Then these are the very active cells in the brain. So but the hypoxia much effect on these not activated microglia. But some of the microglia actually shifted to during the hypoxia. You see here all the capillary, you can see the same place, but some of the microglia is shifted. If you look, you can change the color of each day, plate and diesel, and green and red. The yellow indicates the same place, but you see here the local microglia is shifted to little bit. These are all same place over the 1 week. So some of the microglia actually shifted to move. So what's happening? If this is here, the capillary appear, new capillary appear here, and you see the start of the angiogenesis, the microglia actually seems to guiding. So it's over the time, we took the 6 hours, 8 to 12 hours. Here, the new capillary coming and then connecting to this area. So you see the microglia covering this point. And after that, the capillary normalized, the microglia is gone. So that indicate the microglia also participate in this capillary restructuring. So the final part, what else affect capillary blood flow is the blood viscosity and the deformability of the red blood cell and the cell surface layer are important components. Yes, actually, these are all affect the capillary blood flow, but in chronic stage, there are cell-to-cell interaction between endocells with microglia and pericyte may be involved in this structure of the brain capillary networks and thus the long-standing baseline capillary flow. So here the restructure in the capillary, there's a microglia is actively participated. This is the final part of my talk. Thank you very much for your listening. Thank you.
Thank you, Dr. Masamoto. That was a great talk. And I see audiences today probably have a lot of questions. So if you have any questions, you can write down in the text message, or you can freely speaking and ask a question to Dr. Masamoto. Okay, we have one question. Would the hydration change the viscosity in addition to the hemocrit and the hydronidase? I can see very well. Reduce the viscosity.
We didn't measure the viscosity in the dehydrated blood, but within the image, you can count it how many cells. So then, if we register blood, that's relatively increase in the blood cells. So if you increase the blood cells, that means the hematocrit increase and also viscosity increase. So that basically the capillary resistance high in dehydration condition. But the point is the fluctuation is more important to uniformly deliver the blood cells. If the fluctuations low, then only the same route is the blood cells going in. That is my data showing. Is that your question? Answer to your question?
Professor Zhang,
Yeah.
is that you can maybe speak to?
Yeah, sorry,
yeah.
I skipped that point. So thanks for the special
No, thank you.
resolution. We use the for structure imaging, we use a 0.2 micrometer, but it takes a long time. For functional stage, we use a 0.5 micro per pixel, and resolution is slightly low, but we compensate based on the intensity profile and also average over the 20 trials. So we have a compensate the sub of the sub-pixel resolution. We believe.
Okay.
That's the limitation of the capillary. Yeah, cell-to-cell interaction variance. Good microglia response to injured patient. We didn't. Yeah, good point. Yeah, we didn't actually measure the condition of the pericyte. That's the next part we need to look at. That why the microglia is attracted to the angiogenesis location. That is yeah, we're missing that point. So good, it's a good question. So some of the study during the information that there can maybe there signal from the blood may attract to make microglia, but we didn't see any status of the pericyte. Sorry.
Okay. Is there any other questions from the audience? I can see that many students are listening today, and
right.
Yeah.
Can I ask a question?
Yeah.
So it's a Kazu. It's
Hey.
great talk. So I think question is I actually asked about special resolution. So I still getting into the point there. So because you have a 5 micron diameter in the sense in the capillary, 0.5 micron you have special resolution. That means you actually have like you are in typically 10% of diameter change is going to be quite big in the capillary. So a lot of cases are probably 5% changes. So question is, what is the detectability coming down? Because you said, okay, statistically significant or not, there is all depending on your detectability of your measurement.
Yes, it's a naive point. I know it, but we are very careful about that accuracy of the image calculation. Sorry, I have a exact slide here, but the how do we measure capillary diameter? So
you look at the profile, I suppose. You look at the profile, then you filter it. You basically you have a kind of a modeling. That's
right. Based on the intensity profile. Yeah. This is a lecture image here. So the resolution about 0.2 micrometer here. This is the steady state condition. So if we use sorry, I didn't have functional. Yeah, here functional activation. This is a image shows, and then we took the intensity profile along the perpendicular to the axis of the capillaries intensity profile, and then we make the threshold to find out the exact diameter. To do that, we fit the intensity profile using the Gaussian functions here. So that means compensate the pixel resolution. And we rotated these the lines to find out the minimum width along this the inch point of the capillaries.
Okay.
So yeah, you're right. So that's we need to be very careful to calculate, because the intensity also influence, and sometimes red blood cells shade the capillaries of the plasma markers. And we use filtering these intensities fluctuations before the image analysis. And then, yeah, that's why we use a single trial, we use a single trial measurement. We also statistically measure based on the distribution of these diameter at each point along the 80 trials.
Mm-hmm.
And one more comment for this measurement. We need to be also careful about the shift in the focus. It's a very two-photon microscopy is very weak to the changing in the focus. That we need to accurately calculate the diameter. We need a Z information. That is
so
also
you got breathing. I mean, there is animal kind of breathing. A lot of times, actually, so it's like it's moving slightly up and down. Just what you said, like
right.
Somehow it's focusing the change. Somehow there is
yes.
So that
yes.
have to be very careful.
Yes, that is we always cautious about the changes in the focus, and we always look at the intensities along the capillaries, and if the intensities are the outlier, then we cut out the data, and then if the activation also cause the blood volume increase, that is also the shift to the focus at the activation point area.
So did you, okay, just one last question relevant to the so did you actually trying to synchronize with more like a breathing pattern, breathing cycle? So like I used to, when we were in the Pittsburgh, we used to try to synchronize, trying to minimize a defocusing problem.
Right. You're right. Yeah, that is another way to overcome the shift in the focus. But animal in the mice's respiration is so very fast, and if it's possible in the rat, but scanning time is much slower than the respiration rate, and also that the change is due to the respiration is still within the fluctuation, within the noise. So if we can increase the number of trials, that is compensated because of the random noise.
Okay.
Thank you.
Okay, Kazuto, we have one more question. Just Jihan from Sungkyunkwan University, he has a question. So in chronic hypoxia, microglia involved in the formation of a new capillary.
Right.
Do you have any predictions on which receptor or molecule of microglia is involved in this phenomena?
Thank you. Yeah, right. Yeah, the answer is no. Sorry, we need a pharmacological experiment to answer that question. We only know the literature basis, but I'm not sure about that. Signal cascade is also involved in the hypoxic response. Yeah, somebody shows the VGFC or kind of gap junction type of the proteins.
Kazuto.
Yes.
Maybe I can ask some questions. Thank you. I think there are too many data. Sometimes I got confused.
Sorry.
But I have maybe two questions. First one is because you show this data of sensory stimulation at 148 Hertz and in different order. I wonder what's what was the how to interpret the data. Do you find differences of this capillary blood flow changes with respect to different frequency of sensory stimulation, or maybe no?
Thank you. Why we use the different frequency? Because of we need to modulate the neural activity more gradually. So if we compare only the two conditions, it's we cannot compare the neurovascular capillary in effectively. So we like this one. If we including resting state, still the firing in the neurons. So the 1 Hz, 4 Hz, 8 Hz, maybe different neurons activated, but they are basically these activation is variable over the trial. So we ranked sorted based on these neurons activities. Then we can more effectively compare these one by one in activities. That is the reason why we use a different frequency here.
Okay. Yeah, thank you. And while maybe this is not your data, but you showed a no reflow is relevant to the obstruction by neutrophils.
After stroke?
Like no reflow phenomenon is by neutrophil. I think that was a Switzerland group data. Yeah. So I wonder if like if this from this title, like when you deplete neutrophil, then does the author see no reflow phenomenon is resolved or disappeared? It's just curious.
I guess, yeah, they did some experiment after the injection of the anti-neutrophil, but I didn't know here. We didn't see any data here, but only the TPA. But yeah, other paper also showed on the Alzheimer's disease condition. Also, this antibody is effective to recover the capillary flow. Yeah, that's the reason why, because the kind of inflammation condition, there may be the more activated in the leukocyte, and the increase in the probability to stuck in the capillaries. So if we delete the leukocytes, so the more flow is more smooth. Yeah. Only we show this dehydration condition also same. That's because the leukocyte is more big compared to the red blood cells.
Dr. Sung-ji Kim also has another question. Do you think whether capillary velocity change is more important than capillary diameter change for the localization of activity?
For mapping purpose, or
Sung-ji, maybe you can ask. You can speak.
Well, you actually basically emphasized about all the diameter changes. You know, I kind of I'm wondering when you actually the mapping purpose. So you trying to localize precise activity. I think one of the figure you have those one. So it's a blood flow seems like they're widespread. Then there's some of it's like metabolic changes are very well localized to the because like I mean risk are better there. So question is you are measuring two different indicators. So which might be the better way to localize precisely to the neural active site, like neural active barrel those.
Right. Yeah, it's a kind of our question, right? It's a but physiological, but the capillary. I don't know. The time to change the capillary is very small. As I showed the earlier part, most of capillary diameter change we cannot detect. So how to measure volume?
Well, you measure.
Correct?
You measure capillary diameter changes.
Yeah, but it's informative for mapping purpose. We need to know the activation pattern here. So actually, we are trying next to stimulate optogenetically in the area of the neurons. It's very small, it's the population.
So let's assume that because assumption is you have incredibly high, like resolution 2-photon system. I mean, you just get theoretical purpose also, right? So you optogenic stimulate very few cells. Or like you look at the nearby capillary. And then it's like, then you look at diameter measurement, also you do the velocity measurement. So hitch is more widespread compared to the neural active area. It's like optogenic active site. You know, there was like Albert did something like similar experiment using the product volume, so optical imaging. So if you do 2 photon, you can look at it very precisely. Then that may be like very interesting. It's a fundamental issue.
Agree. But yeah, right, yeah. Yeah. If the capillary diameter change is high enough, then we can map this. But my impression, it's always fluctuate the diameter, but change due to neuron activity is still small, not coding the neurons information here. But velocity is more likely it's due to the caused by the blood cells flow. So certain point of the capillary diameter is regulated, effective to the change, cause the blood flow change. But if the higher than the diameter of the red blood cells, doesn't matter. No, the capillary diameter fluctuation, no much effect on the flow velocity. So no information from the neurons activity. So yeah.
Hachu, did you also measure the red blood cells velocity changes during seizures? Because I can see there's a huge difference in capillary diameter changes when you compare the sensory stimulation and seizure case. But this is actually related with the Sungji's questions, like which one is more sensitive to the neural activity. So have we ever measured the velocity changes during seizures?
Let's collaborate.
Yeah.
Not yet. Yeah,
Okay.
if we use this rat animal model, so we can see the both information together, and then see how this fluctuation affect on the flow. It's important to know. I agree. Thank you.
Okay. So I think we have many wonderful questions.
Thank you.
And Kazu, thank you so much for this like remarkably wonderful seminar. And I think I learned a lot, and I hope all the audiences today from Korea learned a lot from your seminar.
I missed to visit the Seoul.
Yeah.
Oh, we need to get you here,
Yeah, I really missed.
Yeah.
First, I got a mail. I can go to the Seoul. But you said online.
So next time you should come.
I visit.
Okay, well, maybe you can invite him.
Not just he's not going to come until we invite him. Okay.
Thank you.
Okay.
Yeah.
Thank you so much.
See you soon. Yeah. Thank you very much.
Bye.
Yeah. Before we finish, why don't we take a picture all together?
Yeah, sure.
Yeah, please show